So children, coming to the structural characteristics, it says here the structural characteristics contribute to gas transport. Now if we look at the structure of an erythrocyte, the side view, if we see, it looks something like this, right? Now if it looks something like this, it says here this is the biconcave shape, right? So biconcave shape means there is a concavity, you can see a concavity or a depression on both the sides. If you look at this, this gives a biconcave look, right? It is concave, or concavity or a depression is found on both the sides. This also gives a look of a dumbbell, right? Now what happens is that this gives a huge surface area for oxygen to be carried. So what happens is that the mature red blood cell does not have a nucleus, right? So it is not nucleated, there is no nucleus that is found in mammalian RBC, right? So this helps to accommodate maximum amount of hemoglobin, and it is the hemoglobin only that is able to carry oxygen, right? So and other than that, if we talk about the structural characteristic, other than the absence of nucleus, which provides more accommodation for the hemoglobin to carry blood, right? A maximum accommodation of hemoglobin to carry blood, this helps. Other than that, we can see that it does not have mitochondria also, right? So energy of course is required. So ATP production is anaerobic, that means energy is produced, but oxygen is not utilized for that, right? So then what happens is that the oxygen that the hemoglobin is able to carry, it carries full amount of oxygen, because no molecule of oxygen is used for ATP production here, right? So the concave surface of the mammalian RBC is helpful in increasing the surface area. So that is one very important thing, and it is superb example of complementarity of structure and function. Structure and function here means that the kind of structure it has, it complements or it helps to carry out the function easily. That means all the functions that the erythrocyte is carrying out, that is basically they contribute to the gas transport. The structure of the RBC is good enough for the RBC to carry out its function properly and to the best level. So the mammalian RBC is without the cytoplasmic organelles. Like the mitochondria, even the Golgi complex is not found inside. Endoplasmic reticulum is also not found. Ribosomes are also not found, right? So that's about the structure and how the structure helps in the contribution of its function, right? So it carries the structure, it increases its biconcavity, increases the surface area so much that it can carry around 97% of hemoglobin, right? So if you look at the structure of the RBC, this is the side view, which is around 2 micrometers, and the top view, which is around 7.5 micrometers, right? So that's the cell structure. If we come to the function of the erythrocytes, the first thing is that the formation of erythrocytes, we know it takes place in the adults in the red bone marrow. So formation, the process or the formation of the erythrocytes is known as erythropoiesis, right? So formation is known as erythropoiesis. So that is how, this is the process, this this is the process by which the RBCs are formed. In mammals, erythrocytes are formed in the red bone marrow, particularly present in the ribs, sternum, pelvis, etc. that I told you, right. So if we come to the function of the erythrocytes, the main function of the erythrocyte is RBCs are dedicated to respiratory gas transport. Now what is the respiratory gas transport? Transport of oxygen and carbon dioxide, that is the respiratory gas transport. Hemoglobin binds reversibly with oxygen. Now if we look at the structure of the hemoglobin, right. So hemoglobin structure, globin composed of, it is if we talk about hemoglobin, let's break up the word first. Hemoglobin, here it is made up of two parts, the heme part and the globin part, right. So globin is composed of 4 polypeptide chains, which is 2 alpha chains and 2 beta chains, right. So now if we look at the structure of hemoglobin, right, so we will see here that it is made up of 4 polypeptide chains, right. Number 1, 2 alpha chains, 1 alpha chain, second alpha chain, 1 beta chain, second beta chain. So it is made up of 2 alpha chains, these are polypeptide chains, right. So 2 alpha and 2 beta chains, right. You can see here 1 and 2 alpha chains, 1 and 2 beta chains, right. So that is the 4 polypeptide chains. Then it says it is made up of heme, which is a pigment bonded to each globin chain, and it is this heme part that gives blood the red color, right. So heme is the pigment which is bonded to each globin chain. Let's look at the structure now. You can see here, these are the polypeptide chains, right, and this structure here, this is the heme part, right, which is attached to all the 4 polypeptide chains, and it is this that gives color to the blood, the heme part which gives color. So attached to each polypeptide chain, we can see here there are 4 heme that are there. Next it says here, heme central iron atom binds 1 oxygen, right. So heme central iron atom. So if we again look at this structure, 4 heme, this is the central iron atom, right. It binds 4 oxygen atom. So this would be 1, 2, 3 and 4 oxygen atoms. That means 1 molecule of hemoglobin is able to carry 4 molecules of oxygen, right. So 4 iron attached to the heme carrying 4 oxygen atoms. Then it says here, each RBC contains 250 million hemoglobin molecules, right. So 1 RBC, so if I draw an RBC like this, 1 RBC, this carries 250 million hemoglobin molecules, right, and then each hemoglobin molecule carries 4 oxygen atom. So if you multiply 250 by 4, it will come to 1000 million oxygen atoms that it will carry, right. So you can imagine 1 RBC can carry almost this much of oxygen atoms, right. So that's about the structure of hemoglobin. Then if we talk about the normal values of hemoglobin, males have 13 to 18 grams per 100 ml of blood, whereas females have 12 to 16 grams per 100 ml of blood, right. So females have a little lesser hemoglobin, right? So that's the hemoglobin structure, right? Hemoglobin helps in oxygen loading in the lung. So when hemoglobin is in the blood, right, oxygen comes in the lungs, and when the hemoglobin comes in contact with the oxygen, it produces oxyhemoglobin, which is ruby red. Ruby red means it is bright red, right? Ruby red means it is bright red. Then oxygen unloading. Oxygen loading in the lungs means in the lungs the hemoglobin gets loaded with oxygen, and when it gets loaded with oxygen, it forms oxyhemoglobin, right? Then the blood carries oxygen to the tissues where it gives away the oxygen. So unloading of oxygen takes place in the tissues. So this produces deoxyhemoglobin or reduced hemoglobin, which is dark red because it then takes carbon dioxide also, right? Carbon dioxide loading in tissues, 20% of carbon dioxide in the blood binds to hemoglobin, which forms carbaminohemoglobin, right? Then every cell has a definite lifespan, right? So also the red blood cells. So the lifespan of mammalian erythrocyte is around 120 days, or we can say 4 months. Then what happens after that? After 120 days, what happens to the red blood cells? A breakdown of RBCs, because they need to be destroyed, right? So breakdown of RBCs takes place, and this breakdown of RBC is known as hemolysis. Hemo is a term related to blood. Lysis, splitting. So splitting of the RBC takes place. Where it takes place? It takes place in the spleen, bone marrow and liver, right? And the most important site of RBC disposal is the spleen, and so the spleen is known as the graveyard. It is known as the graveyard because here it is destroyed, the RBCs. Iron, now what happens is destruction of RBC is taking place, it is getting disposed of in the spleen, right? Then it is made up of iron. So iron released by hemolysis, where once that is split, it breaks up into iron. So what happens to the iron? Iron is retained in the body, right? It remains in the body, right? And it is reused in the bone marrow to form hemoglobin. So it is reused to form hemoglobin, right? Then what happens is biliverdin is formed from the protein part, right? What is hemoglobin made up of? It is made up of an iron part and a protein part, that is globin, right? So biliverdin, now what happens to the globin part, the protein part? Biliverdin is formed from the protein part of erythrocytes, and it is almost completely reduced to a yellow pigment, right? That yellow pigment is known as bilirubin, right? So yellow pigment is formed. Let's talk about once again the complete function of RBCs. Exchange of gas, transport of oxygen in the blood from lungs to all the cells of the tissues. They assist with the transport of carbon dioxide from the tissues to the lungs. They play an important role in regulating the acid-base balance of the blood, and so prevents large changes in pH. They also assist when a blood clot is formed, right? So that's about the functions of the RBCs. That's all for today's class. Thank you.